Welcome to the deep dive. I want you to think of a resting cell in your body as like a highly regulated, high-security biomanufacturing factory, right?
Yeah, that's a perfect way to picture.
Right, and it operates on this strict set of internal blueprints. The cell membrane is this heavy-duty security perimeter. And for decades, the rule was that if you wanted to change what that factory makes, you had to physically break in.
Exactly. You had to get past the membrane, navigate all the way through, and deliver new genetic material like new blueprints straight to the core.
But our mission today is to uncover how these tiny cellular bubbles, called exosomes, are completely upending that idea. We're pulling from a really fascinating stack of sources today.
We've got some cutting edge stuff, data from the ACR journals, the International Journal of Advanced Research, and some deep genetic databases on the TGFBR1 receptor.
Yeah, and what they show is just wild. We're tracking a mechanism where exosomes can completely transform a quiet resting cell into an aggressive, heavily armored factory. And the kicker, they do it without transferring a single strand of genetic cargo inside.
Which is, it's staggering for how we understand biology. We've always assumed exosomes were just cargo ships.
Yeah.
Just delivery trucks, smuggling DNA or RNA into other cells to change their behavior.
But instead, it turns out they don't need to drop off any internal cargo at all. Going back to that factory analogy, they aren't smuggling blueprints inside. They're basically just walking up to the outside wall of the factory, pressing a very specific button, and the whole factory instantly retools itself from the inside out.
Right. It's a radically different mechanism. It's a completely cell-free paracrine signal based entirely on a surface-level handshake.
Okay, so to really unpack how this button pressing works, I think we first need to look at what these exosomes actually are, right? Because our view of them is really shifting.
Oh, absolutely. So exosomes are these tiny extracellular vesicles. We're talking 50 to 100 nanometers in diameter. They're released when these things called multivesicular bodies fuse with a cell's plasma membrane.
And we find them everywhere, right?
Yeah.
Like in all body fluids.
Yeah, exactly. Blood plasma, saliva, breast milk, you name it. But historically, the literature has been obsessed with what's inside them.
Right, the cargo.
Right. Researchers spent years cataloging all the messenger RNA, the microRNAs, the DNA packed inside the bubble. The whole assumption was that the exosome's only job was to protect these fragile nucleic acids while they floated around in the body.
But if you actually look at the membrane of the exosome, the outside of the bubble, that whole delivery truck idea kind of falls apart.
It completely falls
I agree.
apart. Because if it was just a protective shell, it would be a smooth, inert lipid bilayer. But it's not. It's heavily studded with these potent active growth factors directly on its exterior.
We're talking about things like tumor necrosis factor alpha, epidermal growth factor, and the big one for today's focus, transforming growth factor beta, or TGF beta.
Yeah, TGF beta is the key here. The fact that it's sitting right on the surface proves that exosomes aren't just vehicles. They mediate biological functions by directly engaging receptors on the target cell.
Wait, so just to be clear for you listening, the active ingredient isn't hidden inside the bubble. It's actually stuck to the outside of it, like, I don't know, like spikes on a burr.
That's a great visual, yeah. Like spikes on a burr, they carry the signal right on the membrane.
But that brings up a really big mechanical question. If you have this potent molecule, TGF beta, just hanging out on the outside of the exosome, navigating the body's fluids, wouldn't it just get ripped off or degraded?
You'd think so, right? It can't just be floating loosely on the surface. It has to be physically anchored. And the research actually identifies the exact anchor.
Yes, the beta-glycan anchor.
Exactly. It's a transmembrane proteoglycan called beta-glycan. It sits right in the exosome's lipid bilayer and holds onto the TGF beta molecule, keeping it tethered and fully functional on the outside.
And the way the researchers proved this was just brilliant. They didn't just guess. They basically took molecular scissors to the exosome.
Right. They used this treatment called pervanidate, which basically cleaves surface proteoglycans. They aggressively cut the beta-glycan anchor.
And what happened when they cut it?
The detectable levels of TGF beta on the exosome just plummeted, like by 40 to 50% instantly.
Wow. Which means half of the TGF beta wasn't inside the bubble at all. It was tied to the outside the whole time.
Exactly. And the craziest part is once they cut that anchor, the exosomes completely lost their ability to transform the target cells. The internal cargo did nothing. The entire transformation relied on that external tethered TGF beta.
Okay, so let's unpack this. The exosome is essentially using beta-glycan like a grappling hook to hold on to TGF beta on its surface. It's cruising around, and then it bumps into a resting fibroblast. So what is the button it presses? What's the lock for this key?
The lock is a specific receptor on the target cell called TGFBR1. That stands for transforming growth factor beta receptor 1.
And according to the genetic databases, that receptor is located on chromosome 9, right?
Yep, chromosome 9.
Okay.
And functionally, it's a serothionein protein kinase.
Okay, so a kinase. That means the moment the exosomes tethered TGF-beta docks with this TGFBR1 receptor, it flips a switch.
A huge switch. It transduces a signal from the cell surface straight into the cytoplasm without the exosome ever actually entering the cell. Specifically, it triggers what's called SMAD-dependent signaling.
Right.
So it completely bypasses the need for donor DNA. The signal travels from the surface right down to the nucleus.
And we literally know this is the exact pathway because researchers tested it with a chemical inhibitor. I think it was SB431542.
Yeah, SB431542. It physically blocks the TGFBR1 receptor.
Right. They blocked the lock. So the exosomes were still bumping into the cells. The TGF-beta was perfectly attached. But because the receptor was jammed, the whole transformation process just stopped dead in its tracks.
It's an elegant, closed-loop proof. If you cut the anchor, the signal fails. If you jam the receptor, the signal fails. It's all about that surface-level handshake.
So we know the lock and the key now.
Mm.
But what actually happens to this resting cell, this fibroblast, once that button is pressed?
It's a dramatic awakening. The quiet fibroblast is totally reprogrammed into what we call an activated myofibroblast.
And this isn't a subtle change, is it?
Not at all. The cell undergoes this massive structural reorganization. The expression of a specific protein alpha-smooth muscle actin, or alpha SMA, just absolutely skyrockets.
It basically gives the cell physical strength, right? Like it builds stress fibers.
Exactly. The data shows the alpha SMA doubles in just 24 hours. It peaks at day 3 and stays elevated for at least 2 weeks. It turns this passive cell into a mechanical force generator.
Wow. But it doesn't just change on the inside, right? Because there's also a SMAD-independent effect that builds up an external armor.
Yeah, this part is fascinating. The activated myofibroblast starts synthesizing massive amounts of hyaluronic acid HA. It builds this thick, highly hydrated protective coat around the entire cell.
And they proved this with that erythrocyte exclusion assay, which sounds incredibly technical, but it's actually really visual.
It is. Basically, they drop red blood cells into the dish. Because the hyaluronic acid coat is so thick and electrically charged, it physically repels the red blood cells.
So you just see this clear, empty halo around the cell where nothing can touch it.
Right. And to prove it was HA, they added hyaluronidase, an enzyme that eats hyaluronic acid. The halo completely disappeared.
That is so cool. So it's basically a heavily armored, highly contractile factory now. But this brings up a really crucial detail from the sources. What happens if you just dump regular free-floating TGF-beta onto the cells without the exosome?
That's the million-dollar question. If you use soluble TGF-beta, you still get the alpha SMA spikes in the armor. The receptor still fires.
Okay, so the exosome isn't strictly necessary for that part.
Right, but, and this is a huge but, only the exosome-tethered TGF-beta could trigger the fibroblast to produce significant amounts of fibroblast growth factor 2, FGF2.
And FGF2 is a big deal, right?
A massive deal. It's a primary driver of angiogenesis, which means it tells the body to rapidly build new blood vessels and remodel tissue. Free-floating TGF-beta couldn't do that.
Wow, so the context matters. The exosome itself is physically clustering the receptors or maybe using co-receptors to change how intense the signal is.
Exactly. It's a much more sophisticated set of instructions.
So bringing this back to you, the listener, what does this all mean for your health? If an exosome can turn a quiet cell into an aggressive armored factory, pumping out new blood vessels, is that a good thing or a bad thing?
Well, it's a double-edged sword. It completely depends on where the exosome comes from. This exact same pathway can be a weapon for a disease, or it can be this profound tool for healing.
Let's start with the bad news first. How do diseases use this?
Cancer is the primary example. Solid tumors, like in breast cancer, actually hijack this exact cell-free signaling pathway. The cancer cells secrete these exosomes to manipulate the local healthy fibroblasts around them.
So the tumor is basically outsourcing its own construction. It forces the healthy cells to build a fibrotic fortress around it.
Exactly. By forcing the fibroblasts to become myofibroblasts and pump out hyaluronic acid and FGF2, the tumor creates a shield against the immune system, and it gets a brand new blood supply to feed its own growth.
And the sources mention that this gets even worse under physical stress, right? The cycle stretch data.
Yeah, that part is terrifying, but brilliant. As a tumor grows, it gets crowded. It experiences mechanical stress, literally cyclic stretch. The research shows that when breast cancer cells feel that physical stress, they dramatically increase the number of exosomes they spit out.
Oh, man. So the more crowded the tumor gets, the more exosomes it fires off to recruit more fibroblasts to build more space. It's a vicious cycle.
It's a highly efficient pathological loop, all driven by this surface-level handshake.
Okay, so that's the dark side. The tumor uses this cell-free signal to build a fortress. But on the flip side, if we know how the button works, we can press it ourselves for good.
Exactly. And this is where the regenerative medicine data comes in. The sources look specifically at stem cells from the umbilical cord Morton's jelly mesenchymal stromal cells, or WJMSCs.
Right, but they don't just grow these stem cells normally. They put them under hypoxic conditions.
Yeah, severe low oxygen, just 5% oxygen. It sounds bad for the cells, but it actually mimics the environment of early embryonic development, or like a severe wound site.
It stresses them out so they release the good stuff.
Exactly. It stabilizes a protein called HIF1 alpha, which forces the stem cells to release exosomes that are just incredibly potent. They are densely packed with regenerative signals.
And the logistical beauty of this is that they're just vesicles, not live cells.
Yeah.
The research showed you can freeze them, cryopreserve them, and they don't lose their shape or their active surface signals.
Right. You just thaw them out and the exosomes are ready to go. And when researchers applied these hypoxia-primed exosomes to human dermal fibroblasts, the healing cascade was unbelievable.
Yeah, the transcriptomic data showed they massively increased fibroblast proliferation, and they specifically upregulated the CO1A2 gene.
Which is huge because that gene encodes for type I collagen. That's the primary structural protein for your skin and connective tissues. The exosomes are directly commanding the cells to rebuild the physical scaffolding.
Well, without transferring any DNA, just pressing the buttons. But here's where it gets really genuinely mind-blowing. It's not just about wound healing, it's about aging.
Yes, cellular senescence. This is the hallmark of aging. When your cells stop dividing, they get stuck, and they just sit there releasing inflammatory toxins that degrade the tissue around them.
And to test this, the researchers looked at a specific aging biomarker, right? Beta-galactosidase.
Yeah, beta-galactosidase builds up heavily in these old, stagnant, senescent cells. But when they treated these aging cultures with the stem cell exosomes, specifically at a 1 to 10 or 1 to 30 dilution, the beta-galactosidase activity significantly dropped.
Wait, really? So it's literally reversing the biochemical markers of cellular aging?
Yeah, it's either waking the cells back up and restoring their function, or it's clearing them out to stop the inflammation. But either way, it is actively reversing senescence markers. And again, all through surface-level paracrine signaling.
It's just a complete paradigm shift.
Yep.
We went from thinking exosomes were basically dumb cargo ships smuggling nucleic acids to realizing they are these hyper-sophisticated signaling platforms.
Absolutely. They use anchors like beta-glucan to present active ligands. They engage receptors like TGFBR1, and they trigger everything from collagen synthesis to tumor angiogenesis without ever breaching the cell wall.
It's incredible. And for you listening, this isn't just abstract science. Understanding this mechanism is literally the frontier of personalized medicine. It's paving the way for cargoless therapies that target fibrotic diseases, heart failure, and aging itself.
It gives us a completely new framework. If we can target these surface anchors or synthetically replicate these exosomes, we can disrupt cancer networks or initiate profound tissue regeneration with unprecedented precision.
Which fundamentally redefines how we look at biology. And, it leaves me with this one lingering kind of terrifying thought.
Oh, yeah. What's that?
Well, if we now know that exosomes can fundamentally reprogram a cell's entire fate just by shaking hand with the right sequence of external receptors, what else is doing this?
Oh.
Right. Could there be unknown viruses or pathogens out there that have already evolved to hijack this exact button-pressing mechanism? Like, what if they're triggering devastating systemic diseases purely by tapping the outside of our cells, and we've just been stubbornly looking for their genetic material on the inside this whole time?
That is a terrifying but entirely plausible possibility.
Something for all of us to mull over. Thanks for joining us on this deep dive, and we'll catch you next time.